Good morning, everyone. I'm Dr. Saif, and today we are tackling a topic that's not just biochemically elegant, but also clinically critical. We are talking about something called isoenzymes, those clever molecular twins of the enzyme world. We start with a picture. Picture this, it's 2 a.m. The monsoon season is going on, and there's a lot of rain. And a 50-year-old gentleman from a nearby town, maybe his name is Mr. Sharma, he's rushed into the ER. And he's complaining of crushing chest pain, which is radiating to his left arm. These are classical symptoms, right? Now your senior resident immediately suspects a myocardial infarction, which is a heart attack. But how do you confirm it with biochemical precision, especially when you need to differentiate it from a severe muscle ache or muscle strain from heavy weightlifting, or maybe even a bad liver infection due to some contaminated street food? This is where our stars for today, isoenzymes, come into play. They are like the specialized detectives of our body, each leaving a unique signature that tells us exactly which tissue is in distress. They catalyze the same reaction, but they have distinct properties, identities, and even locations. So let's peel back the layers of this fascinating topic and see how these molecular variants, which are isoenzymes, give us such crucial insights. Now, simply put, the first question arises, what are they actually? They are basically different molecular forms of an enzyme that catalyze the same reaction. But here's the kicker. They differ in their primary, secondary, tertiary, and even quaternary structures, along with kinetic properties like Km and Vmax, regulatory mechanisms, and even tissue distribution. Think of them like different models of the same car brand, like a Maruti Swift, a Dzire, a Brezza. All are Marutis, all get you from A to B. But they have different engines, different features, and are optimized for different uses or terrains. Even our isoenzymes are encoded by different genes or arise from alternative splicing of a single gene. Now that we know what they actually are, the next question is, why do we need them? Here I want you to imagine your heart. It is constantly pumping, needing steady aerobic energy, right? Now imagine your skeletal muscle. It is capable of explosive anaerobic bursts during a sprint or lifting a heavy rock or anything. Isoenzymes allow the body to fine-tune metabolism to the specific needs of each tissue. It's biological efficiency. Now here we come to a quick distinction, mainly between isoenzymes and allozymes. Isoenzymes are different forms in a single individual. Now this can occur due to different genes or splicing. Now example of this is LDH1 in heart and LDH5 in liver. These are classical examples of isoenzymes. But allozymes are different forms within a population from different alleles at a single gene locus. Example, different variants of G6PD in different individuals. So here I want to give you a cricket analogy which might help. Isoenzymes are like having a cricket test team and a T20 team. Different compositions for different formats, but the same sport. Allozymes, on the other hand, are like having two different players from different states in the same position on the same team, which is slight genetic variation. Okay, now that we have some kind of intuition about these isoenzymes, what they are, why we need them, let's come to a couple of examples. There are several isoenzymes, but we are going to talk about the big players. The first one is LDH, which is short for lactate dehydrogenase enzyme. LDH catalyzes the reversible conversion of pyruvate with NADH to lactate. Now more about LDH. It is actually a tetramer, which means it has 4 subunits. Now these 4 subunits are made of mainly 2 types of subunits. This is the H subunit and the M subunit. This is easy to remember because H is predominant in heart and M is predominant in muscle. Now coming to the 5 isoforms. This is LDH1, LDH2, LDH3, LDH4, and LDH5. So LDH1 has all 4 H's. LDH2 has 3 H's and 1 M. LDH3 has 2 H's and 2 M's. LDH4 has 1 H and 3 M's. And LDH5 has 4 M's. Now all these M subunits will be found in the skeletal muscle and liver. So that is for sure. LDH1 is most specific for the heart. As you can see, it's not very hard to remember this. Now let's talk about a couple of differences. LDH1 is more specific for the heart, and one small caveat is it has high affinity for lactate and is inhibited by high pyruvate. It mainly favors lactate to pyruvate conversion. It doesn't favor the opposite conversion. In the highly aerobic heart, it helps convert lactate to pyruvate, which is then going to enter into the Krebs cycle for efficient ATP production. It ensures NAD+ is regenerated for oxidative dephosphorylation. Think of it as the diligent batsman who ensures the score is kept steady. LDH5, on the other hand, is most common in muscles or liver. It has lower affinity for lactate, and it mostly favors the pyruvate to lactate conversion. Its role is more in skeletal muscle during intense exercise, example, maybe a kabaddi match. When oxygen is limited, LDH5 is rapidly going to convert pyruvate to lactate. This regenerates NAD+, which is crucial for glycolysis, and this produces ATP anaerobically. Now in MI, there is something which we can look for, LDH flip. Normally, LDH2 will be higher than LDH1. But what happens in MI is there's a flip because LDH1 is more found in the heart cells. This leads to increased LDH1 in the blood. And this has been found to peak 2 to 3 days post-MI. Although this is superseded by more specific markers now, as we have seen in our cardiac markers video. And LDH5 points to liver disease, example hepatitis, cirrhosis, etc. And LDH, and it is observed that in lung diseases or lymphomas, LDH2 or LDH3 are raised. So that covers the clinical significance of the various LDH isoforms. Let's now go to the second example, that is CK. No, it's not Calvin Klein. It is creatine kinase. It is the rapid energy buffer. CK catalyzes the reversible transfer of phosphate group between creatine and ATP. Now this phosphocreatine acts as a rapid energy reserve, especially in tissues with high and fluctuating energy demands. It quickly regenerates ATP from ADP. Think of it as your phone's power bank. It is ready to charge when the ATP dips. Now the structure of CK is a dimer, which means it is made up of two main subunits. And the two main subunits here are M subunit and B subunit. In LDH, we saw the H and the M. Here we are seeing the M and the B. The M subunit is, of course, present predominantly in the muscles. And the B subunit is present predominantly in the brain. Now, there are three major isoforms. There is CKMM, CKBB, and CKMB. CKMM has two subunits of M, and it is mainly present in skeletal muscles. CKBB has two B subunits and is predominantly found in the brain, along with the smooth muscles. CKMB has one M and one B subunit and is predominantly seen in the cardiac muscle. So this CKMB has a high clinical relevance because since it is present in the cardiac muscle during myocardial infarction, this is raised and it is very specific. Its levels rise between 4 to 6 hours. And we have seen this in the cardiac biomarkers video. And the peak is at 18 to 24 hours. Everything returns to normal by 2 to 3 days. It is mainly a very rapid rise and fall. It is mainly a marker which is going to rise and fall very rapidly. So it's an excellent early marker. Now, a couple of other conditions is CKMM is elevated in skeletal muscle damage. Now this can include excessive exercise or trauma, muscular dystrophy, rhabdomyolysis, and CKBB is elevated in brain injury. This includes stroke, head trauma, or some cancers. But it's very rare to detect this one in the serum due to the blood-brain barrier. It usually doesn't cross the blood-brain barrier. Now that we have covered the CK, let's move to the next one, Hexokinase and Glucokinase. Now both of these catalyze the first committed step of glycolysis, which is phosphorylation of glucose. Glucose plus ATP gives glucose 6-phosphate plus ADP. And as you know, if you recall, this is the first step of glycolysis. This is catalyzed by these two enzymes. Let's first talk about the structure of hexokinase. This has 4 major isoforms, and these are 1, 2, 3, and 4. Now these 1 and 2 forms are called true hexokinases, and these two are present in most of the tissues. This hexokinase 4 is actually called glucokinase. These three forms, 1, 2, and 3, these are regularly present in most tissues like muscle and brain. They have a very low Km value. If you recall about Km values, if they have a very low Km value, it means they have a very high affinity for glucose, which means they efficiently grab glucose even when its concentration is low. Think of a hungry student quickly picking on any food item available in the canteen. And this has a feedback inhibition in the sense that if glucose 6-phosphate increases, the enzyme slows down. This prevents unnecessary glucose phosphorylation when the cell's energy needs are met. Now, glucokinase is mainly present in the liver and pancreatic beta cells. It has a high Km value compared to the first three, which means it has lower affinity for glucose. And it only becomes significantly active when blood glucose levels are high. It's like a fine dining restaurant for glucose. Only opens when there's a flood of people waiting. And one specialty here is that it is not inhibited by the glucose 6-phosphate. There's no feedback inhibition like we saw in the first three. It can keep phosphorylating glucose even when glucose 6-phosphate levels are high. Now, if we talk about the role, the liver acts like a glucose sensor. After a meal, when blood glucose spikes, glucokinase rapidly phosphorylates excess glucose, and this channels it into glycogen synthesis or fatty acid synthesis. It helps the liver buffer blood glucose. Pancreatic beta cells are the primary glucose sensors for insulin release. High glucose makes more glucose 6-phosphate, and this leads to increased ATP, and this leads to closure of potassium channels, which leads to depolarization, calcium influx, and insulin secretion. So glucokinase has a very important role in insulin secretion. In fact, mutations in glucokinase can lead to something called MODY2, or which is short for maturity onset diabetes of the young type 2. It is a mild form of diabetes because the pancreatic beta cells become less sensitive to glucose. And if they get less sensitive to the glucose, then the insulin secretion will decrease. Let's now look at regulation and control. Now, these isoenzymes actually have multiple layers of control. You'll see what I mean. The first one is transcriptional control. So genes for different isoenzymes are switched on or off in specific tissues or at different developmental stages. They are controlled by transcription. Next is allosteric regulation. Different isoforms respond differently to activators or inhibitors. So one example of allosteric is how we can see that glucose 6-phosphate is getting easily inhibited by hexokinase, but it's not getting inhibited by glucokinase. The third one is substrate concentration. Now, due to varying Km values, isoenzymes react differently to changing substrate levels, and this allows tissues to prioritize glucose usage or storage. So the Km values have an effect on how the various enzymes respond to the changing substrate levels. Then there are developmental changes. Think of a baby's metabolism versus an adult's. Isoenzyme patterns change to meet these evolving needs. That is also how they are changed. Now I want to give you a couple of mnemonics here. The first one is LDH. So here you can remember H is for heart and high affinity for lactate and high oxygen. M is for muscle, and it has two major subunits, right? H and M. M is for muscle, more lactate produced and more anaerobic. Another way to remember is that LDH1 and LDH5. My heart has heaps of healthy cells. This is LDH1. But my muscles make more lactate. So in the first one, my heart has heaps of healthy cells. So a lot of H's. And the second one, my muscles may make more lactate. So more M's. So that is LDH5. Then we have CKMB, or you can think of it as short form for cardiac killer myocardial blocker. Or another way you can remember this is cardiac muscle breakdown for MB. Now for hexokinase versus glucokinase. So Hexo is like the ever hungry dhaba owner. He serves food to anyone, which is any tissue, even if there are few customers, and the food is glucose here. So even with low glucose, he serves the food. Glucokinase, however, is like the 5-star hotel manager. He only opens the buffet when there are many wealthy customers, which means very high glucose has to be present for it to begin. Now finally, let's come to the summary. So what are isoenzymes? They are different forms of an enzyme catalyzing the same reaction but with distinct properties and tissue distributions. Then what are a couple of examples? There is LDH, there is CK, there is Hexokinase and Glucokinase. LDH, if you recall, is a tetramer and the subunits are H and M. It has 5 isoforms. LDH1 is present mainly in the heart. LDH5 is present in the muscle or liver. LDH2 is usually more than LDH1, but in certain circumstances, LDH1 is found to be more, and this is seen in myocardial infarction. This is called the flipped pattern. Then we have CK. CK is a dimer and it has 3 isoforms. CKMB is a CKMM, CKBB and CKMB. CKMB is a key MI marker and CKMM indicates muscle damage. Then we have hexokinase versus glucokinase. Both phosphorylate glucose, but hexokinase has very low Km value and it is easily inhibited by glucose 6-phosphate and it is present abundantly. While glucokinase has a high Km value, it is not inhibited by glucose 6-phosphate and it is present mainly in the liver and the pancreas. And finally, we saw how using these isoenzymes, we can diagnose MI, liver disease, muscle injury, or diabetes. And isoenzymes are mostly used in diagnostics to come to the root cause. Thank you for listening to this session. Hope you got valuable insight from this and hope to see you again in the next one. Thank you for watching.